All right, so now that crossing over is complete, let's go through the rest of prophase 1. So what else happens in prophase 1 is the nucleolus disappears, the nuclear envelope disappears, and our chromosomes condense into chromatid. So that's comparable to what happens in mitosis. The only difference is we have crossing over. Now, let's go to metaphase 1. Now, this is where we are again different. In metaphase 1, what happens is our chromosomes line up in the middle. Now, the way they line up in the middle is our homologous chromosomes line up in the middle. So what we're doing is we are first separating our homologous chromosomes. We are not separating by sister chromatids. We are separating homologous chromosomes. Now, what will also happen is our lovely little spindle fibers will attach. So they will attach to each of our homologous chromosomes as so. So this will happen to all of these. Now, in anaphase, what happens is that the homologous chromosomes, this would also have a spindle fiber attached, the homologous chromosomes get pulled to opposite sides of the cell. So again, in anaphase 1 of meiosis, homologous chromosomes are pulled to the opposite sides of the cell. Now, in telophase 1 of meiosis, our spindle fibers break apart, the nucleolus reforms, the nuclear envelope reforms, and our chromatid decondenses into chromatin. Actually, cut that out. Okay, so now what you'll notice is that we started with 6 chromosomes, but now each cell only has 3 chromosomes. So what I'm going to do is focus on what happens in meiosis II. So at this point, keep in mind we have 2 cells that are each haploid. So what's going to happen in prophase 2 is we're going to have the traditional what happens in prophase, which is nucleolus disappearing and the nuclear envelope disappearing. Now, in metaphase, what's going to happen is this is going to be similar to mitosis. So in meiosis 2, or meiosis part 2, metaphase is our chromosomes are going to line up in the middle, and the centromeres are going to attach to each side of the sister chromatid. So that is going to happen in both the cells, because if you remember, at the end of meiosis 1, we produce 2 cells. So I'm going to quickly just kind of make the spindle fibers attach. So this is going to happen here, this is going to happen here, this is going to happen here, and it's going to happen in our other cell where these attach. It's a tight space, and I'm trying to get to this quick, so that is why I'm not giving each one spindle fibers, but imagine if they had spindle fibers attached. So in anaphase, what happens is the spindle fibers will pull our sister chromatids to opposite ends of the cell. So, like in mitosis, in anaphase 2, our sister chromatids are separated. That happens again in both cells. Now, in telophase, our chromosomes unwind, the nuclear envelope reforms, and the nucleolus starts to return. And so what you have is, after cytokinesis, we will have 4 cells that each have 3 chromosomes. If you remember, we started with 1 cell with 6 chromosomes. Now we are making 4 haploid gametes. If you remember, gamete is the fancy way to say sex cell. And each one of them are genetically different. If you notice, they all have different variations in the chromosomal structure. So they have different alleles on each of the chromosomes. And this is what happens in meiosis.